I will present the innovative randomization methods that will be used in the endTB trial, in which we will evaluate multiple regimens for MDR-TB treatment. First, we will start to see what is the Bayesian adaptive randomization and see how it can be applied to the endTB trial. So what are the objectives of a clinical trial? So it's to evaluate new therapies and show their efficacy and good safety profile, to produce rigorous data, and if positive results, then it's to change the standard of care, to put patients on better drugs in a timely manner with a smallest yet adequate sample size. For this, there is a gold standard, which is a randomized clinical trial. You will test experimentals versus placebo or control when it's possible, depending on the context. We have seen for the Ebola trial it was not possible. And then you will randomize your patients by assigning them by chance to one of the groups to minimize the selection bias. This randomization can be simple. You just flip the coin. It can be blocked when you balance the sample size between groups. It can be stratified when you balance the sample size on baseline covariate, or it can be adaptive. And when it's adaptive, it's part of the adaptive design family. When you plan well-defined changes in the design during the trial based on data observed during the conduct of the trial. So when it's an outcome or response adaptive randomization, then good outcomes in a group lead to more patients randomized to that group, and it's a play-the-winner approach. Then you have two different approaches, the frequentist or the Bayesian. To schematize, it's like a bet on horse races. You have two different possibilities. One, you make assumption before the start and you watch the race. And at the end, you test your hypothesis with a type 1 error, which is usually of 5%, which is the probability to have the success by chance. And this is a frequentist approach. Or you have a previous information. You can maximize it and update with observed data. Then you calculate a posterior probability of success. No test is performed, and this is a Bayesian approach. And if you pull them together, the Bayesian approach and the adaptive randomization, then you get the Bayesian adaptive randomization. You start with your prior information, even if it's slightly informative. You randomize your first patient. The first time it's balanced within treatment groups. Then you perform interim analysis on early endpoints. Early because depending on your accrual rate, you want to have time to make some changes in the design before they are all randomized. You calculate the posterior probability, which is a probability of success given the data. Then you unbalance the allocation probability for the next patient using this posterior probability. You update the prior, and once some more patients are randomized and you got the data on the early endpoint, you will perform interim analysis. Then you can easily implement some stopping rule for futility or for toxicity. If it is for futility, then you can stop your experimental group if the posterior probability of having a success given the data is lower to a boundary that you will have defined. And you can also stop for toxicity if you have a kind of an incidence that you don't want to be greater than a limit that you will define. Well, if the posterior probability of this incidence is greater to a boundary that you also define for toxicity, then you can stop your experimental group. So it's a flexible design. It may make the study more efficient, of a shorter duration, potentially fewer patients, and it allows the evaluation of several experimentals in one trial compared to traditional design. So it can be of benefits to many trials. It has been already in oncology, and now it will be for the multi-drug resistant tuberculosis, which is caused by mycobacteria resistant to isoniazid and rifampicin, the two main first-line anti-TB drugs. The current regimen is of long duration, complex, poorly effective, toxic, with a poor adherence, and expensive. Over the past 40 years, only two new drugs recently developed, the bedaquiline and the delamanid, with interim WHO guidance, which have been developed. So there is really a potential for novel drug regimens to shorten the MDR-TB treatment. Few clinical trials are ready to start or ongoing. And from them, there is the MSF initiative with 2 clinical trials sponsored, the endTB and the TB practical. In the endTB trial, we will evaluate 5 experimental regimens, all oral, containing either the bedaquiline or the delamanid, of 9 months duration, compared to the best standard of care of 20 months. The primary endpoint is a favorable outcome at 60 weeks, which is approximately 15 months. It's a non-inferiority design using a Bayesian adaptive randomization based on 2 preliminary outcomes. The final endpoint being too late at 50 months, we prefer to adapt on early outcome, which are the 2 months mycobacterial culture conversion and the end of treatment success, which are positively correlated with the final outcome. So some simulations were performed to explore the benefit of the method before to use it for the endTB, and also to compare the Bayesian adaptive randomization with other methods, and to calculate the sample size to get a fixed power under hypothesis on treatment response design, and perform sensitivity analysis to evaluate the robustness. Here are the results for 1,000 simulations performed on the experimental regimen, either with a non-effective arm and effective arm. So it is in the hypothesis of having one superior arm. So with the adaptive randomization, the median number of patients will be approximately 264 for the effective arm, and for the non-effective arm, it will be 91 patients. When for the balanced randomization, it will be 134, whatever the effective or non-effective arms. So more patients are randomized in the effective arm, and less patients in the non-effective arm. On this graph, you can see the probability of having a true positive result, which is a power, depending on an overall sample size, with the treatment success either of 65% or 75%. The solid line is for the adaptive randomization, and all the dashed lines are for other methods, balanced randomization with or without interim analysis. So when the treatment success is of 65%, for a given power of 80%, for example, then you can see that you will need much less patients with a Bayesian adaptive randomization compared to the other methods. But then if your treatment success increases, then the difference between the power first increased a lot, and the differences between methods is smaller. The sensitivity analysis were done to evaluate the impact of changes in the parameters. We forced a 4-week delay to get the result on the preliminary endpoints, percentage of misrandomized patients during the conduct of the trial, and also change the expected treatment response. And it appears that there is a lower sensitivity to erroneous assumption with the Bayesian adaptive randomization than with the other methods. So there are quite some advantages of using this adaptive randomization. First, the sample size is reduced, with more patients treated with regimens more effective, and this method is quite robust. But if you made some error in the prior assumption, or if you misspecify the controls, then the gains will be limited. And there are also some challenges, because you will need to collect your data almost on a real time and perform frequent interim analysis during the conduct of the trial. Thank you, and thank you to the partners.